Welcome to Microscopic Marvels. All right, today we're jumping into a topic that is an absolute favorite in both theory exams and vivas, the VDRL test. Now, VDRL stands for Venereal Disease Research Laboratory. It's what we call a non-treponemal serological screening test, which is a fancy way of saying we're looking for signs of an infection in the blood, but not for the bug itself. And the infection we're screening for is syphilis, which is caused by that corkscrew-shaped bacteria, Treponema pallidum. It's so common it's even called a standard test for syphilis, or STS. So you might be wondering, how does this test actually work? What's the magic behind it? Let's break down the core principle, because once you really get this, everything else about the VDRL test just falls into place. Okay, for this test to work, you need two key players. First up, the antibody. The VDRL test is on the lookout for something called reagine antibodies in the patient's blood. These are usually IgG or IgM. Now, the key word here is nonspecific, and we'll come back to why that's so important. Second, we need an antigen to actually find these antibodies. And for this, we use a cardiolipin antigen, which, fun fact, is extracted from beef heart. This antigen basically mimics some of the fatty stuff on the surface of the syphilis bacteria. Now let's pause and clear up something that trips up a lot of students, that word reagent. You see, in the context of syphilis, the reagent we're talking about is an IgG or IgM antibody. It gets triggered when our own cells get damaged and release lipid bits. That's what VDRL finds. But you've probably also heard the term reagent in allergies, right? Well, that's a completely different antibody. It's IgE, and it's triggered by allergens like pollen. So please, for your MCQs, do not mix these two up. So the actual reaction happening on this slide is called flocculation. The easiest way to think about it is as a special kind of clumping. When those reagent antibodies in the patient's sample meet the cardiolipin antigen we add, they grab onto each other and form these visible clumps. And those clumps have a name, floccules. So simply put, we mix the two together and look to see if this clumping party happens. Okay, enough with the theory. Let's get our hands dirty, virtually of course. How do we actually do this test in the lab? And more importantly, how do we figure out what it means? The procedure itself is pretty straightforward. First, you take a tiny amount, just 50 microliters of the patient's serum. You have to heat and activate it first, and you put that on a special slide. Step 2, you add 1 drop of our VDRL antigen. Step 3, you pop it on a rotator and mix them for exactly 4 minutes. And finally, and this is super important, you have to read the result under a microscope at 10x magnification. You cannot see this with your naked eye. So those 4 minutes are up. You take the slide and you're peering down the microscope. Now what? What exactly are you looking for? How do we read the results? Well, it's pretty black and white, really. There are 2 things you can see. If the test is reactive, which means positive, you'll see those medium to large clumps, those floccules we talked about. The antigen and antibodies have found each other and are all clumped up. On the other hand, if the test is non-reactive or negative, you'll see, well, nothing. Just a smooth, even solution. No clumping at all. Great. So we know how to do the test and how to read it. But why do we even bother? Why is the VDRL test still such a valuable tool for doctors today? Let's talk about its clinical applications. It really comes down to 3 main uses. First, it is an amazing screening tool. Why? Because it's cheap. That makes it perfect for testing huge groups of people, like an antenatal screening for all pregnant women. Second, it's fantastic for early detection. The antibodies it picks up appear just 7 to 10 days after the very first sign of infection. And third, and this is a big one, it's absolutely crucial for monitoring treatment. The antibody levels, or titers, actually go down as the patient gets better. Okay, everyone, listen up. This next point is extremely high yield for your exams. I practically guarantee you'll see this somewhere. While we usually do a VDRL on blood, we can also do it on cerebrospinal fluid, or CSF, the fluid around your brain and spinal cord. And here's the kicker. A positive VDRL test on CSF is considered diagnostic for neurosyphilis. That means if you see it, you've confirmed the infection has spread to the central nervous system. It's a huge deal. Now, the test isn't equally good at all times. Its sensitivity, its ability to catch the disease, changes with the stage. In the primary stage, it's pretty good, about 82% sensitive. But in secondary syphilis, when the infection is raging, it's a rock star, 100% sensitive. It will not miss it. As the disease moves into the late stage, though, its power fades a bit, dropping down to around 72%. Let's circle back to that monitoring part because it's just so important. We love the VDRL for this because those antibody levels directly tell us if the treatment is working.
After a patient starts therapy, we want to see their titer, their antibody level, take a nosedive. We're looking for at least a fourfold drop by the third or fourth month. That's our signal that the antibiotics are doing their job.
But, of course, no test in medicine is perfect. And for your exams, you absolutely have to know the limitations and pitfalls of the VDRL. So let's dive into what can go wrong. Okay, picture this. Your examiner in Aviva asks you, can the VDRL test be wrong? Can it ever give you a false positive result? Your answer should be a very confident, absolutely yes. This is what we call a biological false positive, or BFP. Remember how we said the reagent antibodies were nonspecific? Well, this is why that matters. Other conditions can also damage our tissues and cause these same antibodies to pop up. Things like leprosy, malaria, or even just being pregnant can give you a false positive. A simple way to remember these is a mnemonic. My poor liver has really bad reactions. For malaria, pregnancy, leprosy, hepatitis, relapsing fever, BFP, and reactions. Now for the other classic pitfall, the prozone phenomenon. This one's a bit of a mind bender. It's when you get a false negative result, not because there aren't enough antibodies, but because there are too many. Imagine tons of antibodies swarming every single antigen molecule. They get in each other's way and can't link up to form those clumps. It's like having too many people trying to get through a doorway at once. They just create a jam and nobody gets through. So what's the fix for this weird prozone situation? It's actually incredibly simple. All you have to do is dilute the patient's serum. That's it. By adding some saline, you reduce the antibody concentration, giving them enough elbow room to finally link up and form the floccules we're looking for. Easy fix.
Phew. Okay, we've covered a lot of ground. So let's boil it all down to the absolute must-know facts. These are your one-line revision notes to lock in right before your exam.
So here it is. VDRL is a non-treponemal slide flocculation test. It detects nonspecific reagent antibodies, your IgG or IgM. It is diagnostic for neurosyphilis when you test the CSF. It is fantastic for monitoring treatment because the titers will fall. And finally, always remember the two big pitfalls, biological false positives and the prozone phenomenon. Got it? Good.
If you like this video, then please like, comment, and subscribe.